Hello and welcome to today's lecture on immunity to viruses. I just want to remind you that most of the figures that will be used during this lecture can be found in your Janeway book, and at the bottom of the screen you'll have references to them if you want to check them for yourself in the book. So as you know, pathogens come in a great variety of different sizes and have different lifestyles. And they can generally be grouped into a variety of different categories that you see here. And our immune systems have evolved to battle these different types of pathogens very differently. And the immune responses against these pathogens, as you've learned, can be both from the innate side or the adaptive side. And all of these work in consortium based on the level of the antigen and the amount of the microorganism to try and alleviate the host of the infection. Now, today we're going to be focusing on immunity to viruses and the different immune responses that we've evolved to battle viral infections. Now, this lecture will build on your knowledge of various different immune responses and how they work together to eliminate pathogens such as viruses. Throughout this lecture today, we're going to be covering briefly what viruses are, what their lifestyles are, what differences between viruses are. We'll then cover innate immune responses to viruses, as well as adaptive immune responses to viruses. And then we'll talk about how viruses have evolved to evade and subvert our immune systems to try to create a long-lasting infections. Now, before we get into the complexities of the immune responses and specific ways that viruses act to counteract our immune systems, let's talk about a little bit of virology and what viruses are, and then how we can classify them. And you'll see why the classification system is important to understanding the immune responses to these viruses. So let's start off with some very basic virology and ask, what is a virus? So a virus is a very small infectious agent that can replicate only inside of living cells of an organism. So this is why viruses are called obligate intracellular organisms. They cannot replicate outside of their host cell. Now, viruses can infect all types of life forms. So viruses can infect mammalian cells, plant cells, and even bacterial cells. However, today we're only going to be talking about the immune responses of mammals. So let's dive into exactly what the structural features of a virus are. So viruses exist as independent particles outside of the cell, and these individual particles, often called virions, contain the genetic material of the virus. They have a protein coat surrounding the genetic material called a capsid, and sometimes they have a lipid envelope surrounding that protein coat. And viruses use host cells as replication factories. Once they invade a cell, they replicate many times and then are released from the cell. And then viruses can be spread to different hosts by a variety of different means, depending on the tissue that the virus infects. So, for example, viruses that infect lungs are often spread by sneezing or coughing, like the influenza virus. And if we look at the very basic viral life cycle, viruses typically enter the cell by a couple different means. They can either do it by membrane fusion, illustrated here. So you can see that the membrane of the virus interacts with receptors on the host cell, and then this outer membrane fuses with the membrane of the host cell to then release the genetic material of the virus into the cell. Viruses can also enter host cells by endocytosis. And this means that, again, the virus interacts with membranes on the outside of the cell, but then this complex is then endocytosed and brought inside. So instead of the membrane fusing with the host cell, as in membrane fusion, the entire particle is endocytosed. Once the virus is in the cell, the virus uses host cell replication mechanisms in order to replicate itself. And this can happen either in the cytoplasm or in the nucleus. And some viruses even have components of replication that happen in both the cytoplasm and the nucleus. The virus will also use the host cell's transcription and translation machinery in order to generate more copies of itself. To exit the host cell, the viruses use three general means. They can either apoptose the host cell that they're in, which will release the viral particles into the surrounding environment. They can use what's called viral budding, where the membrane here, you could see the cell membrane here, and the viral particle here is kind of coming out. So this is similar to the membrane fusion that you saw with viral entry. And you can also have virions leaving the cell via reverse endocytosis, where virions are within an endosome that then comes to the surface of the cell membrane and releases viral particles. So you can see the parallels between viral entrance into the host cell and viral exit. So now that's the very, very basics of virology and how viruses work and replicate in their host cells. So let's go into classification systems of viruses. Now, when you're classifying pathogens, there's a variety of different ways to do it, and different fields prefer to do it in different ways based on what's most important for them when they're studying said pathogen. Now, in virus classification, the most prominently used method of classification is called the Baltimore virus classification mechanism. So this is based on how mRNA is produced during the viral infections. So this is really important because viruses need to generate mRNA from their genomes in order to produce proteins of their own origin and to replicate themselves. Now, these different viruses, as you will see, produce mRNA by different means. And this leads to the fact that there's actually different PAMPs, or pathogen-associated molecular patterns, that different viruses have based on their replication mechanism. And so when we're talking about this viral classification system, keep that in mind and think of potential PAMPs that you hear of as we're going through this as an exercise that we will then confirm the answers to later in this lecture. So each of these groups, as I said, has different PAMPs that are exposed during replication, and some of the viruses have very unique features of their nucleic acids, so that their nucleic acids actually have different structural components compared to their host cell. And some of the viral nucleotides are sometimes in inappropriate locations in the cell. So for example, typically in a host eukaryotic cell, you find DNA only in the nucleus of the cell. And if you, for example, were to have DNA in the cytoplasm, this could be a sign for the host cell that there could be an infection. Now, this is a very general classification based on the Baltimore virus classification system. So we have 7 different classes of viruses that we will be delving into, but in general, they're grouped into DNA viruses, RNA viruses, and reverse transcribing viruses, which I will get into the details of. So let's dive into those. So in our DNA viruses, we have 2 classes. We have class 1 and class 2 viruses. So their genomes, so the difference between class 1 and class 2 viruses is that class 1 virus genomes are double-stranded DNA, whereas class 2 DNA viruses are single-stranded DNA. And most of them replicate within their nucleus. There are a couple notable exceptions, but in general, DNA viruses replicate in the nucleus of the cell. Now, if you remember what I said about DNA predominantly existing in eukaryotic cells in the nucleus, this is some insight into the PAMPs that are available, correct? You wouldn't want too many DNA viruses replicating in the cytoplasm, as this could be a potential PAMP for the immune system to recognize the infection. So single-stranded DNA viruses, seen here, have a double-stranded DNA intermediate that they generate themselves using a polymerase that they bring with them. And so these are the 2 different classes of DNA viruses. And some examples are for class 1, we have herpes viruses and pox viruses. So pox infamously causes smallpox. And some class 2 examples are circoviruses, which have been linked to patients with CNS infections or nervous central nervous system infections, and parvoviruses. So parvovirus B19, which is the most studied, has been linked to childhood arthritis. And that kind of concludes our section on DNA viruses. Now, if we look at RNA viruses, they come in 3 different varieties in class 3, 4, and 5. So genomes of RNA viruses can either be double-stranded or single-stranded RNA. And you can see, so class 3 is our double-stranded RNA virus family. Class 4 and 5 are both single-stranded RNA libraries, and I'll get classes, and I'll get into the difference between the two in a little bit. So most RNA viruses replicate within the cytoplasm. And again, this is a very general rule. There are some exceptions, but in general, most replicate in the cytoplasm. And most of them encode their own RNA-dependent RNA polymerases in order to replicate. So class 4, the difference between class 4 and class 5 is their sense of their RNA molecules. So for example, our class 4 RNA viruses are what are called positive single-stranded RNA viruses. So this means that their RNA is what's called infectious, and that means that their RNA can directly be translated into viral proteins. And in order to replicate and in order to make more of their genomic material, the positive sense single-stranded RNA viruses have to go through an antisense or non-coding single-stranded RNA intermediate in order for that negative sense single-stranded RNA to then be used as the genome to make more positive-stranded mRNA viruses. So for class 5 viruses, these are negative single-stranded RNA viruses, which means that their own genetic material is not called infectious because their RNA cannot be directly translated. In order for class 5 RNA viruses to be replicated or for their proteins to be translated, the single-stranded RNA virus is directly transcribed into messenger RNA, so the negative sense strand encodes for the mRNA, which can then be translated. So again, we have our class 3 double-stranded RNA viruses, our class 4 positive sense single-stranded RNA viruses that go through a negative sense intermediate, and we have our class 5 negative single-stranded RNA viruses. Now some examples. Our class 3 viruses include rotaviruses. Class 4 viruses include hepatitis C, dengue virus, SARS, MERS, and rhinoviruses. And class 5 viruses include Ebola viruses, measles virus, mumps virus, rabies virus, and influenza virus. So RNA viruses are actually one of the most studied and well-known groups of viruses that exist. Our final very general grouping of viruses are our RT viruses, or reverse transcribing viruses, which are also called retroviruses. So what's really unique about our RT viruses is that they utilize a virally encoded reverse transcriptase in order to generate double-stranded DNA that is then integrated into the host genome. So if you look here at the 2 classes of reverse transcribing viruses, our class 6 and 7 viruses, they start as single-stranded RNA viruses or as double-stranded DNA viruses. However, what's interesting is that both of them will integrate double-stranded DNA into their host genomes, and then essentially the host cell can't differentiate between integrated viral genomes or their own, and so the integrated viral genomes are replicated by the host machinery. So what happens here is that our single-stranded RNA for class 6 or mRNA that's generated in the cytoplasm of our class 7 viruses, the single-stranded RNA is reverse transcribed into double-stranded DNA. So transcription, as you may know, is the process from which double-stranded DNA it is transcribed into single-stranded RNA, but in reverse transcription, the reverse happens. So we have single-stranded RNA molecules that are reverse transcribed into double-stranded DNA molecules, and these double-stranded DNA molecules are then integrated into the host genome. So class 7 viruses' DNAs are, as I said, initially transcribed into RNA that is then used as mRNA and as pre-genomic RNA, and this will be the template for reverse transcription. Some examples of class 6 viruses are HIV, most infamously, as well as bovine and marine leukemia viruses and mouse mammary tumor viruses. And class 7 examples include hepatitis B virus. So as I mentioned, these viruses are integrated into the host genome, and if they integrate too closely to an oncogene, this is the reason why they can cause cancer, which is why you see multiple cancer-causing viruses on this list. So now we've gone through a very general classification system of viruses. And I hope you're able to appreciate the nucleic acid diversity that you can see either in the actual virions, so the different strandedness and types of genomic material viruses have, as well as their unique intermediate replication nucleic acids. So now that we have a very general sense of what viruses are and kind of had a brief introduction to virology, let's dive into the immune responses against viruses. And a good place to start is always the innate immune response, because that is also how the first response that happens upon infection. So in innate immune responses today, we'll be going through PAMP recognition of viruses, type 1 interferon production by plasmacytoid dendritic cells, as well as NK cells and group 1 ILCs. So let's start with some PAMP recognition. So in order to recognize viruses by our immune systems, most viral PAMPs, so pathogen-associated molecular patterns, are nucleic acids. So this could either be direct sensing of the viral genome or of genome replication intermediates. And as I mentioned before, some viruses have unique features of their nucleic acids. And viral nucleotides can also be in inappropriate locations in the cell. Now here's a list of various different pattern recognition receptors, or PRRs, that recognize different viral PAMPs. So for example, you can see here that a majority of the pattern recognition receptors recognize a PAMP that is nucleic acids. So for example, we have our endosomic PRRs that recognize double-stranded RNA, single-stranded RNA, and modified DNA. We also have our Rna receptor, which recognizes uncapped RNA with a 5' triphosphate in the cytoplasm, and MDA5, which recognizes double-stranded RNA in the cytoplasm. Not all virus PAMPs are nucleic acids. So for example, if TLR2 is also important in the immune response against certain viruses that have viral surface glycoproteins. So if you can remember from our introduction to virology section, you could see that based on the different ways that viruses exit the cell, they can have different things on their membranes, and TLR2 is also critical in recognizing these modifications. So now we've gone very briefly through different PAMPs that are recognized by viruses, and if you would like more on that, you can go to your section in Janeway that covers pattern recognition receptors. Let's jump into type 1 interferons. So type 1 interferons, or interferons alpha and interferon beta, induce what is called an antiviral state of the host. So, and we'll get into what that means, but it's kind of self-explanatory. It's a state of the body or of the immune system that minimizes the chances that viruses can infect the host and kind of brings up all the safeguards that we have to prevent this from happening. So type 1 interferons are secreted predominantly by leukocytes, dendritic cells, and fibroblasts. So what happens is that these cells will sense a viral infection through PRRs and start producing type 1 interferons. So once type 1 interferons are secreted from the cells that have sensed the viral infection, interferon alpha and beta bind to a common cell surface receptor known as interferon alpha receptor, or IFNAR. IFNAR then uses JAK and STAT pathways. Specifically, it uses type 2 and JAK1 to activate factors STAT1 and STAT2, and these then form a complex with IRF8, which is called ISGF3. And ISGF3 will then go into the nucleus of the cell that is sensing the interferons, and it'll bind to the promoters of interferon-stimulated genes, or ISGs. This suggests that ISGF3 is a transcriptional activator of these interferon-stimulated genes, and interferon-stimulated genes are what induce the antiviral state. So here we can see a summary of this. So once ISGF3 is activated, the ISGs, or the interferon-stimulated genes, will change a lot about the host, including resistance to viral replications, induction of proteins that suppress translation. They increase the presence of MHC class 1 expression and antigen presentation in infected cells, and they activate further downstream cells that are involved in the antiviral response. So if you were to look at viral replication, kind of taking a step back and looking at it from entry to replication to protein generation to exit, type 1 interferons can block essentially every single stage of virus replication, which is really great in the prevention of a viral infection. And the main generators of type 1 interferons are called plasmacytoid dendritic cells. And these dendritic cells, also known as PDCs, are a very distinct line of dendritic cells that secrete large amounts of interferon, up to thousands of times as many as other cell types. So here we could see a schematic representation of the inside of a plasmacytoid dendritic cell. And the reason that they secrete so many amounts of interferon is believed to result from the efficient coupling of TLR-mediated virus recognition and interferon production. So for example, here you can see the endosome of our PDC, which has TLR7 and 9, and you can see that there's a very elegant pathway that signals through TRAF6 and RF5 and 7, which leads to the high production of type 1 interferons. Now, the ability for PDCs, which express lots of different amounts of viral sensing PRRs to generate type 1 interferons is really critical, as the large amount of type 1 interferons allows for the other cells of the body to be protected from infection. What's also important to note is that PDCs also express CXCR3 on their cell surface. So CXCR3 is a receptor for CXCL9, CXCL10, and CXCL11, which are all made by T cells. And the presence of the CXCR3 on the surface of the PDC mediates PDC migration from the blood to the lymph nodes. And this is really important in allowing for the generation of a really strong immune response upon viral infection. Now let's jump to type 1 ILCs and NK cells. So NK cells and group 1 ILCs make up what's called a type 1 innate immune response. So these are very critical in fighting intracellular pathogens, including viruses. So NK cells and ILC1s are both innate lymphoid cells, so they come from the lymphoid lineage, but they are not adaptive immune cells. And they are induced by IL-12. The presence of IL-12 is generated upon the recognition of viruses by PRRs, and they produce interferon gamma. They both produce interferon gamma, and then NK cells also produce other molecules that we will go further into in a little bit. So this is another schematic of this, showing that our type 1 ILCs, 1s, and our NK cells are generated predominantly by IL-12, IL-7, and IL-15, and they both produce a lot of amounts of interferon gamma. And interferon gamma is a cytokine of the interferon family, and it's really important in activating macrophages and allows for increased presence of active macrophages throughout the host body, which is really critical in phagocytosis of viral particles, which we'll get into in a few slides. So